softly spoken, with no strings tying you to me. I'm happy in your company with no emotion.
Morning, class. Today we're going to learn how to use the microscope. It's going to be an adventure for us. When we carry our microscope, we always carry it with two hands. One on this part, which is called the neck or the arm, and one on the base. If we do this, we'll never drop our microscope. These things are expensive and they're very precise. Sadly, I have seen a couple of students drop microscopes over the years and break parts of them. So we don't want that to happen. When we get our microscope, we set it on our desk, of course. We unwrap the electrical cord. Someone's twisted this all around here. Unwrap our electrical cord, plug it in, and we want to make sure our light works. Whoever turned it in last time did not turn the light off. You should turn the light off before you before you return it to the cabinet. We want to turn it so it's facing with its back toward us, so the front end is open. That's because when we're working with the microscope, and of course we're going to use glass slides, we can put slides on easily like this. If it's turned around this way, it's kind of difficult to put the slides on. It's a small thing, but it will make a difference. Our microscopes are used by other students. So when we set up our microscope, we want to take some lens paper. And we're going to use lens paper to clean the lens on the microscope. There are lenses that are much like the lens on your glasses, except they're ground to a different magnification. We're going to take a little bit of alcohol, or you could use alcohol wipes if you're careful with them. We're going to take a little alcohol, we're going to put it on our, oops, we got a lot of alcohol. We're going to put it on this part where our eye will be looking through the microscope. Also on the objective lens, there's a little lens right here. We're going to be wiping these off with just a little bit of alcohol. Now, if we have alcohol left over, we can wipe the rest of our microscope off with it. We would want to clean the knobs on the microscope that we're going to be touching, maybe the stage area that we're going to be touching. Okay, this is to prevent us from getting some kind of eye infection. Okay, so it's always a good idea just to, we don't usually use the lens paper to clean anything except the lens. But if we have, after we've cleaned the lens and we have a little bit of alcohol left on our lens paper, why not go ahead and wipe things down with it? We want to dry these carefully and polish them up with our lens paper. The lens paper will not scratch the lens on the microscope. The lens on the microscope are made out of soft glass so that they can grind it. Just like the lens on our glasses are ground so that they magnify. Our glass slides are made out of hard glass. They won't scratch very easily. So we can use a regular paper towel to clean them with. Okay. We can use a paper towel to clean the microscope itself, like this. Okay. But we never want to use a paper towel on the lens of the microscope because they will scratch. And if you've ever tried to look through glasses that have scratches on them, that's what you would get. Let's go over the parts of the microscope before we get started. Our instructions have a picture of a microscope on it. It has numbers on it, so you could fill these in as we go along, so you can put the parts of the microscope there. Okay, we're going to start at the top. The top of the microscope, we have this part right here, which is called the tube. Obviously, it kind of looks like a tube. Then we have the eyepiece right here, or the ocular. Now this is where the lens of the microscope are found. One of the lenses. We never want to put our fingers on the lens of the microscope because our fingers have oil on them. And that's what the police or investigators take fingerprints with, is the oil on our fingers. If we get the oil on our lens, it collects dust, and after a while, it makes it hard to see things. So we never put our fingers on the lens itself. So we have the eyepiece or the ocular. We have the tube. And then we have this piece right here that turns around and around. This is called the revolving nose piece because it just kind of sticks out in front of the microscope, and it has these objectives on it. These objectives have lens on them also. We never want to put our fingers on that either. This one right here, you see, has a red line on it, and it's got a number 4, which means it magnifies 4 times. This one has a yellow line on it. We call this low power. It has a 10 on it, which means it magnifies 10 times. Then we have high power, which has a 40 on it, which means it magnifies 40 times, and it has a blue line. So we have the red one, the red line, that's just scanning, magnifies 4 times. We have the yellow line, low power, magnifies 10 times, and we have the high power with the blue line, which magnifies 40 times. So how many times does our microscope magnify? Well, the eyepiece magnifies 10 times. All of our microscopes, the eyepiece has are 10x, 10 times. You can get different lens to put in here, which, like, you could get one that was 20, which means that this would magnify 20 times. But how much does our microscope magnify? If we put the scanning objective in place, which magnifies 4 times, we multiply 4 times 10, which is 40. So the scanning objective magnifies 40 times. If you turn the low power objective into place, it magnifies 10 times. 10 times 10 is 100. So on low power, we get a magnification of 100. We turn our high power into place, which magnifies 40 times. 40 times 10 is 400. So when we're using high power, we can get a magnification of 400. Okay, so this is the revolving nose piece, and these are our 3 objectives on our microscope. This part right here of our microscope is the stage. It's like the stage in the theater. It's where everything happens. This microscope has a mechanical stage, a mechanical clips, excuse me. And you can see these knobs control the movement of the stage. So if we put a slide on here, we can use these knobs to very carefully move our slide around. Underneath the stage, we have this disc that turns like this. This is called the diaphragm. And you notice it has different size holes on it. Okay, we're going to show you how that works. The light is down here. If we turn the light on, you can probably see right here that it's lit up. If we rotate that diaphragm, it's no longer lit up. So we have a small hole, a larger hole. Now it's starting to light up a little bit, and there's more light. So let's kind of tilt that like that. Maybe you can see it better. You see, if we rotate it, you get more or less light. It's important to be able to adjust your light when you're looking with the microscope. Okay, we have the base or the bottom of the microscope. Then we have a couple of knobs here. We have the coarse adjustment knob. Look at the stage. The stage moves up and down rapidly with the coarse adjustment knob. Okay. Then we have the fine adjustment knob. It doesn't look like it moves the stage at all. Now the coarse adjustment knob, we never really want to turn the knobs on our microscope a whole lot. We only want to turn them about like this. And the other thing we need to remember is when the stage stops, we stop. We don't want to try to force these knobs, because when it goes, the stage will only go so far down. When it stops, it stops. It won't go anymore. We can twist this if we want to, but it's not going to go anymore. And if we do twist it, there are gears inside of here that are going to get stripped out. And sadly, I've seen that happen too. So we have our coarse adjustment knob. We don't want to move it a whole lot, maybe about that much. And we have the fine adjustment knob. And if we move it, we should never have to move the fine adjustment knob more than one turn up or one turn down. Because if we turn it more than that, it gets the microscope out of sync, and then Mr. Smith has to come along and get it back in sync for you, okay? Let's see, is that everything we have on our microscope? So ocular or eyepiece, 2, revolving nose piece. We have 3 objectives, scanning, low power, high power. We have our stage. We have our mechanical stage or mechanical clips. We have the diaphragm underneath, which adjusts the light. We have the light itself, we have the base, and we have the arm or the neck of the microscope. Now I brought along 2 other microscopes because I want to show you some different parts on them. So we'll set that one right there. This microscope right here has regular stage clips on it. You see them like this. We don't want to lift these stage clips up and bend them out of place. Some students do that. They lift them up too high. All you do is barely slide your slide that you're going to look like at underneath the stage clips like that. Okay, so these are regular stage clips right there. Another microscope that we have with us is this one here. This one here intimidates students. It just looks more complicated, and it is a little complicated. It has a mechanical stage that works from underneath, which is very nice. If you're working with a microscope, you just reach underneath. You can continue looking through the objective and still move your stage back and forth. It has the coarse adjustment knob on the outside, and the fine adjustment knob is right on the inside. But what I want to show you on this is the diaphragm on it. This has a regular iris diaphragm, and it works by turning this. It's very nice. This is a very nice microscope. It's a little bit tricky to focus. That's why it intimidates students. But it also has 4 objectives. It's got a scanning, it has a low power, it has a high power, and now it has an objective that says 100 on it. That means it will magnify 100 times. So if we're looking with the 10 powered eyepiece and the 100 power objective, it's going to magnify 1,000 times. To use that lens, you have to have oil. You have to put oil. You have to have your specimen on here with the cover slip on it. We'll get to that in a minute. And you have to put a drop of oil. And you have to put the end of the lens into the oil. You have to, it's called oil immersion, because the light has to travel up through the microscope. The light starts from our light down here at the bottom, of course. It goes up, it goes through the exact middle of the aperture or the opening on the stage right here, and then it goes up through the exact middle of the eyepiece, up, and through the exact middle of the ocular. And light tends to refract and go out like this. And so what the oil does is it keeps the light from going out like that. If you use the oil immersion lens without the oil, it looks all like the light is fractured and going out in all directions. Anyway, this has a diaphragm on it, which is an iris diaphragm, which is very nice. This is a very nice microscope. Only the best operators of a microscope can use this one because the focusing of it is very precise, and it has the iris down here. And it also has the ability to raise this iris up and down. See this knob here? We can move this up and down, okay? Which is an added advantage if we're looking at something with a microscope. Okay, part C on your microscope, titled Focusing the Microscope. And it says, always follow these steps, do not vary. The microscope is a precise instrument, like I've said. If we don't use it correctly, we can certainly have issues. So if we're going to focus our microscope, first we need something to focus it on. So we're just going to take this little box right here. We're going to take out one of our prepared slides. These slides are slides that are already prepared, and they have the glass slide. They have a cover slip on them that's actually glued on. Now this slide here is muscles, 3 types of muscles. We have smooth, cardiac, and striated muscle. We'll be looking at these later in the year. Okay, but the first thing we do, if we're going to focus our microscope, is we put it on low power. Okay, now we might want to use scanning, but rarely. We will use scanning today. So we put our microscope on low power, usually, that's what we do. We take our slide and we put it on the stage. In this case, I have a mechanical stage. I'll show you how to do it with the regular stage clips in a minute. And we center what we want to look at, we center it. What center means is, like I said, the light goes through the exact middle of this aperture on the microscope. So that's where we have to put the specimen that we want to look at, in the exact middle of the opening on the stage. So it would be right there. So I'm going to move this just a little bit like that, and that should be pretty close to the exact middle, because it's going to go through the exact middle of the aperture, through the exact middle of the objective, through the exact middle of the eyepiece. If we don't do that, we might be off to the left or right or up or down, and we won't see what we're looking at. Okay? So the next thing we do is we take the coarse adjustment and we raise it up like that. Now it stops. It won't hit the slide. It will be close, but it won't hit. Now we simply look through the lens. I need to open up my diaphragm so I can have a little light in there. I look through the lens and I turn my coarse adjustment knob down, and it comes into focus. Okay? Then I turn my fine adjustment to make sure it's perfect. Okay? Now I can see some smooth muscle specimens. If I want to look on a high power, I turn my high power into place, just like that. But I don't turn any knobs. I don't raise it up. Some students are afraid it's going to hit. It looks like it's going to hit, but it won't. If you have the microscope focused on low power, when you turn to high power, it will not hit. Because our microscopes are what called parafocal. In other words, if you have it focused on low power, when you turn to high power, it's still going to be very close to being focused. So on high power, all I have to turn is the fine adjustment. And I turn the fine adjustment up or down a little bit. I change my light because it got darker when I turned to high power. Now I can see it better because I made more light coming to the specimen. It does not take very long to focus a microscope. You can do it in about 20 seconds if you know what you're doing. This microscope here has the stage clips on it. So we're going to take our slide. Now notice how we can just slide things on, because the opening, the part that's open is away from me. If it's like this, it's hard to do that, okay? And I'll show you another little trick in a second about that. So we take this has regular stage clips. We're going to turn our light on. We're going to take our slide, we're going to center it. We're going to put it in the center of the aperture or the opening on the stage. We're going to put our stage clips on, just like that. Now I'm not going to use the scanning lens. I'm going to turn it to low power. I'm going to raise before I even look, I'm not even looking in the microscope yet. I'm going to raise it up at the coarse adjustment, and it's going to stop. I look in, I focus down. Okay, now I don't quite see it came into focus, but I'm going to put my thumbs on both sides of the stage like this. I'm going to barely hold the bottom with my fingers. Now I can move the slide left or right, and I can find exactly what I want to look at with my microscope. I'm going to slide over and to this area right here, and I want to look at some striated muscle cells. Oh, these are nice. Okay. Now I'm going to turn my fine adjustment, and my microscope is perfectly focused. Now here's the little trick I was going to show you. My microscope is focused on high power. I can take this slide off. I can take another slide. This one's bone tissue. I'm going to take it, I'm going to slide it into place. And guess what? My microscope should still be very close to focused, and it is. I turn my fine adjustment just a little bit. I adjust my light because I want to see it a little darker. It's a little too light for me. Turn my fine adjustment and it's focused. So there's a trick that you can remember. Once you have the microscope focused, you can take slides off and put them on, and it should still be very close to being focused, which our microscope is. This is a very nice slide. I'm going to try to show you exactly what this looks like. So this is how it would look when you focus your microscope. I've raised the coarse adjustment so the stage is all the way up. I put my slide on the stage and centered it, and you can see that the specimen we want to look at is there, but it's blurry. We don't see the whole field of vision on this. It's very tricky trying to line up the camera with the microscope, but we see enough of it that we'll be able to see how it focuses. So I turn the coarse adjustment down, and you'll see that it comes into focus. Okay, we try to get it as perfectly focused as we can with our coarse adjustment. You can see how we can go up or down with it. Okay, then we turn our fine adjustment, and you'll notice that the fine adjustment hardly looks like it makes it do anything. Okay, now that's focused on low power, so we're going to turn to high power. Our high power clicks into place, and you notice also there's this pointer inside the microscope that is supposed to be there. Okay, that is supposed to be there. That's part of the microscope. Now we're going to take my thumbs and put it on the slide, and I'm going to barely move it and see how much it moves. Now the pointer is pointing right at some of the striations on the muscle tissue. Okay. Now if I want to take this slide off, I'm going to turn it back to low power. Oops, my camera just moved a little bit. I'm going to slide this slide off. From the front, I'm going to take the bone tissue. I'm going to slide it on under the clips. And there's the bone tissue with the Havasarian canals, which we will study later this year. That's low power. We want to turn to high power. All we have to do is barely turn the fine adjustment, and there comes our Havasarian canal into focus. Okay. So we looked at the Havasarian canals and looked at the prepared slide of the 3 types of tissue. That's how we focus the microscope. It's very easy to focus once we get used to it. That's why we always follow the same steps. In the lab, you want to do everything step by step by step. If you do it step by step, you'll be very happy with the results, and you make less mistakes. Well, class, we set our microscope aside for a second because before we were using prepared slides. These are prepared slides. They're prepared in a laboratory and they're purchased. We could make our own permanent slides, prepared slides, but usually we make what's called a wet mount slide. So the prepared slide right here, like I said, it has a cover slip on it and it has glue on it, a special type of glue on it that holds it in place. That's prepared slide. So we're going to be making a wet mount slide. To make a wet mount slide, first thing we want to do is we want to make sure our slide is clean. I'll just dip it in some water and we wipe it off with a paper towel. Okay? We just use regular water. Now, the reason we can do this is, that's right, the prepared slide is hard glass, okay? And it won't scratch. We don't want to use the lens paper on the glass slide, or we don't want to use it on our cover slip because the lens paper is expensive and it's made for the lens on the microscope. So we dip it in, we take our piece of our little tiny cover slip, and now we take a small piece of paper towel, something like this. We get our little cover slip wet and we just use our thumb and one finger, index finger or this one here, to clean it with. And we just rub it around like that, and we hold it by the edges after we get it done like that, so we don't get fingerprints on the cover slip or the slide. Now I'm just going to set it like that for a second so I can pick it up easier. The reason we use one finger and thumb to clean our cover slip with is this: if we put two fingers like that, or if we just hold it like this, we will very easily break this cover slip. I'll show you, I'll demonstrate. If we put our cover slip like that and we're using two fingers, listen very carefully, you heard it break, okay? And it's very easy to break, okay? If we break one, we throw it away, that's okay, but we don't want to waste the cover slips by throwing them away, and we don't want to break them. You know, they're not real expensive. We can buy 1,000 of them for $5, but if everybody breaks one or everybody throws one away, then pretty soon they're gone because they get used up rapidly. The most important reason we don't want to break them is because we want to be experts at using the microscope, and experts try to do things correctly. Okay, so our instructions say, cut out a word from the want ads in the newspaper that contains a letter E. So we cut one out from the newspaper, and I'm going to put it right there. Whoops, it flipped over. That happens sometimes. I'm going to put it right there, okay? Try to get it. I'm going to use my cover slip. I don't want to put my fingers on my slide, so I'm going to use my cover slip. I want to get this thing turned because I'm supposed to put it on the right side up, okay? Using the dropper, I want to put one drop. It flipped over on me, so I'm going to have to flip it back over. That happens sometimes. There we go. All right, and I'm going to put my cover slip on. When we put the cover slip on, I always hold the cover slip out like this. I've seen a lot of students hold them backwards like that. To me, that's very hard to do. But I've got big giant fingers, so you can do it either way. But we need to touch the edge of the water like that. So it goes right along the edge of the slide, and then we lower it down as close as we can get it, and we let it go. And what that does, when the cover slip goes down like this, it forces the air out and we don't get air bubbles. A lot of times students will say, Mr. Smith, I've got this thing in here, what is it? And I look and it's this big round purple thing, and it's very beautiful. It's an air bubble. And then they're all sad because it's an air bubble. So that's how we make a wet mount slide. Now I might have a little water on the bottom, so I'm going to take my little paper towel like that. I have a little extra on the side right here. I'm going to take my cover slip and pull it off like that, okay? Another technique that's good to know, and we'll show you how we're going to do this. We're just going to lift that up. We're going to put a piece of white paper on it so you can see this, okay? We can use stains. This is methylene blue, and we'll use methylene blue in a week or so when we do a lab where we use the microscope and we look at the different kinds of cells, cheek cells. There's other types of stain like iodine, which we use on plant cells. Okay, we just take a drop and we put one drop on this one side of the cover slip right there. And look how it's going underneath. You can see it going underneath, right? So then we take a little piece of paper towel and we put it on the opposite side. Like this. And you can see how it's pulling the purple stain underneath. And we just take it, we move it a little bit more, and the purple stain gets pulled underneath the slide, the cover slip, okay? And it's going to stain whatever in there. So when we look at the letter E, it might be stained a little, we might see a little purple stain in there, okay? That's a little technique that's very good to know. Okay, we have our microscope back, and we want to take our slide that we made, our wet mount slide, and look at it. So we're going to take it, we're going to turn the microscope to low power or scanning. We're going to take the stage, we're going to lower it down so we can get our slide on, and we're going to center the letter E. Now the letter E is right side up as you would read it. Now you can see right here where the light is shining on it. Okay, I'm going to turn like this. Hopefully you can see that. The light is shining up from below right on the letter E right there. So it should be pretty close to centered. Okay, we're going to raise our stage up with the coarse adjustment. We're going to look in the eyepiece. We're going to focus down. And there it is, perfectly focused. We're going to turn to low power, which is going to magnify it 100 times. Now I'm only going to have to turn the fine adjustment a little bit. I'm going to put my thumbs on the side of the stage and my fingers barely, I don't want to hold real tight, just like this, so I can move my letter E so it's exactly in the center of my field of vision. I can turn to high power. Now I'm only going to turn the fine adjustment like that. I'm going to move it again just a little bit. Turn back to low power. Okay, I'm going to turn back to high power. Check it with my fine adjustment with the high power. Okay, so I want you to see how this is done, so we're going to show you that also. Now remember, I put my letter E on the stage right side up. Okay, you can see obviously it's upside down. Okay, it's not focused yet. We have it on the scanning. I'm going to focus it down with a coarse adjustment, and you're going to see it come into very good focus. So you can see the pointer pointing right at the middle of the letter E. Okay, so the microscope, guess what? It turned my letter E upside down and it turned it backwards. I want you to notice this. If I put my thumbs on the side of the slide and I push it to the left, it went to the right. If I push it left, it goes to the left. If I push it up, it goes down. I push it down, it goes up. So the microscope reverses and turns everything upside down. That's because of the prism and the lens that are inside of it. Okay, we're going to turn to low power, which is going to magnify it 100 times. We're just going to click it into place. We're going to turn the fine adjustment. Excuse me, we're going to have to use the coarse adjustment. We're on low power. Barely turn it like that, okay? Now you can see the field of vision is the area or the circle that we see with the microscope, okay? I'm going to turn my light a little darker. This is something you have to learn how to do with your microscope. If it's too bright, you have to adjust your light a little bit. I like it right there. I can see my letter E much better, okay? So I'm going to turn to high power. And I'm going to have to adjust my light again, because when you turn to high power, you see something bigger but less light comes in. And I'll show you why in a second. Maybe I'm not going to have to. All right, let's try focusing here a little bit. Go back to low power. Get her focused perfectly. Okay, now we're going to turn it to high power. And we'll find our letter E. There it is. Okay, see where the pointer's pointing? Right at the cross part of the letter E. We don't see all of it. So on high power, we do not see all of it. We see a very tiny part of the letter E. So if we're looking on high power, we see a very small part of what we're looking at, but it's magnified much greater. So if we turn back to low power, we see much more of the letter E. Okay? We see much more of it, but it's not magnified as much. If we go to the scanning lens, we see all of the letter E, completely all of it, but we see it's not magnified nearly as much. So the higher magnification you go, the less of the object that you see. Now see where the pointer is pointing? Okay, the pointer doesn't move, the pointer stays. So if we turn it from there to low power, it's still pointing at the same place. If we turn to high power, I'm going to focus just a little bit. We turn to high power, it's still going to be pointing at exactly the same place, but we see much less of it. Now your instructions say, draw 2 circles in your lab book, like those below. Draw the letter E and circle A. Okay, so you're going to need, instead of 2 circles, you're going to need 3. You're going to need one for the scanning, one for low power, and one for high power. And so you're going to draw the letter E just like it looks on scanning, low power, and high power. So scanning would be magnified 40 times, low power would be magnified 100 times, and high power would be magnified 400 times. Measuring with the microscope. We took a ruler, a little metric ruler, and we placed it on the stage, and it's on the scanning objective, which means it's magnifying 40 times. We put the edge of it so we can see it, and we can see the marks. Each mark on the ruler is 1 millimeter. Okay, and so we're going to move this like this, so we can see that the scanning objective is 1, 2, 3, and a little bit millimeters across. Each millimeter has 1,000 micrometers. So the distance across our scanning objective field of vision is 3,000 and we're going to say 3,100 micrometers across, or 3.1 micrometers in diameter. We need to be able to measure with our microscope. We need to be able to tell how big something is that we're looking at. And to do that, we need to know the diameter of the field of vision on our microscope. When we look at our microscope, we see a circle. How far across is the circle? If we know the diameter of the field of vision, we can use that to figure out or determine, estimate the size of something that we're looking at. So to do that, it's really quite simple. We take a ruler. This has a metric side to it. We place it on our stage and we center it so the edge of it is visible and we can see the marks on our ruler. Okay? Then we know how far across it is. To determine the size or the diameter of our field of vision, it's really not that hard. We look in the eyepiece. That's not a very good circle. Let's try that again. And we see our letter E. Okay? And our letter E on 40 magnification looked to be about that big. Okay? And so we drew our letter E. This is why you were told to draw the letter E, so that we could estimate the size of it. So it looked like that. Okay? Now we know that we measured it. It was 3.1 millimeters across on scanning lens objective. That equals 3,100 micrometers. And the symbol for micrometers is like UM, but the U has a tail on it like that. That's the symbol for micrometers. So we know it's 3,100 across from here to there. So how big is our letter E? This is on scanning, which magnifies 40 times. So to figure out what is the diameter on low power, we take the scanning is 40 and the low power magnifies 100 times. So we divide 40 into 100. We drop that off. 4 goes into 10 2 times. That's 8. 2. 4 goes into 25 times. 2.5 times smaller. The high power is 2.5 times smaller. So we take 3,100 and we divide it by 2.5. Okay? That's going to be 1. That's going to be 2.5. We have to move that decimal over, don't we? So we have to add a 0. Okay? Okay, we're going to subtract. That's going to be 6, 0, 2.5. 25 goes into 60 2 times. That's going to be 50. You get 1, 0. 25 goes in 100 4 times. Okay? And we bring down 25 goes into 0, 0. So low power, the diameter is 1,240 micrometers. What's it going to be on high power? Okay? We take high power magnifies 400 times. we divide it by 40. Okay, that's going to be 10, right? So the high power is 10 times less than the scanning. The scanning is 31, like that. 10 times less. All we got to do is move the 0 right there. So high power, the diameter that we're going to see on high power is 310 micrometers. Okay? That's not a very good symbol for micrometers. Okay? So if we look at the letter E, and on the scanning, it's 3,100 across. The letter E looks to be about half of that, right? So we would say the letter E's diameter, is the size of the letter E is about 1,500 micrometers. So once we know how to measure with the microscope, it will be interesting for us to do a little investigation. We're going to take our slide that we used before, just going to wipe it off. Okay? We're going to take our little cover slip, we're going to clean it, dip it in a little water, and again, very carefully hold it by the edges. Okay? Now our instructions tell us to take a piece of a magazine. So we're just going to take a magazine, and we're going to find a piece of colored magazine. I like blue, so we're going to take a little piece of blue here, okay? We're going to cut it out. I cut a piece out already, and we're just going to set it right on our slide right there. We're going to add a drop of water or two to it. Okay? We're going to take our cover slip. I'm going to touch it so it runs along the edge first, lower it down slowly. And this has got a lot of water on it. It's got more than I like, so I'm just going to take my paper towel like I showed you. I'm going to soak some of that water up just so my slide is more stable. Okay? Okay, so we have this slide, and we have this little piece of paper that has blue on it and a little bit of red. So let's see what this looks like under the microscope. So here's our little piece of magazine, and you can immediately see, if we focus it here a little bit more, get us a little focus going here. That we have dots. We have blue dots, black dots, we have a red line, and we have little tiny pink dots. Now this is the scanning. This is the scanning objective. So let's turn it to low power. Okay, we're going to have to focus it. The dots obviously are bigger. So one thing we learn is the microscope, besides making things bigger, it separates them. You notice how it separated the colors? It separated the black dots from the blue dots and the pink dots, okay? So this is low power, magnified 100 times. High power. Okay, turn our fine adjustment just a little bit, and you see the dots are even bigger. Now your instructions say to draw the low power and the high power. So I would have you draw 3 circles instead of 2 circles. Draw 3 circles. Draw how big they are on scanning. Draw them same color as they are. Draw it on scanning, 40 magnification, 40x, draw it on low power, 100x, and draw it on high power, 400x, okay? Well, class, we've covered a lot of territory today with our microscope. First thing we did was we learned the parts of our microscope: the ocular eyepiece, the tube, the revolving nose piece with our scanning, 40, which magnifies 40 times, 40x, low power, which magnifies 100 times, 100x, the high power, which magnifies 400 times, 400x. We learned also that we multiply the number on the objective times the eyepiece to determine the magnification. We have the stage, we have the stage clips, or we have a mechanical stage. I want to take this slide off. We have the diaphragm underneath, and we have different kinds of diaphragms. This is a disc diaphragm. We have the light. We're going to turn the light off. We have the base. We have the coarse adjustment knob, which turns things very easily, rapidly. We have the fine adjustment knob, which fine adjusts the microscope. And we have the neck or the arm. And we always carry the microscope with two parts, two hands, one underneath and one on the neck. Okay, so when we get ready to put the microscope up, we always take our slide off, we turn the light off, we wrap make sure the stage clips are in, wrap the cord around, and we want to secure the cord. They have these little notches on them there. Just take it around and clip it on like that. Our microscope's ready to go back to the cabinet. Again, one hand underneath, one hand on the neck. Today we also learned how to make a wet mount slide. We learned that the microscope, besides magnifying things, it separates them so we can see more detail. We learned that there are wet mount slides that we prepare ourselves, and we learned that there are prepared slides that are bought, very expensive. They cost about $10 or $11, maybe $12 each. I've seen some cost $18 each just for a little slide. We learned how to measure with the microscope. We use this is a ruler right here. We can measure how big this little green box is with the ruler. And it's the same thing. We use the field of vision, the circle that we see in the microscope, to measure with, just like it was a ruler. It's just a very small ruler. Well, class, we had a very good discussion today of how to use the microscope. If you're patient with it and you work at it and you try to do it as the instructions say, you can become an expert at using the microscope. I had one student come back to me once after she'd graduated. She got a job in Los Angeles at the Los Angeles County Police Department in the forensic department. And when she went for the interview, they said, "Can you use a microscope?" She thought, "Yes, I can use a microscope. Mr. Smith taught me how to use a microscope." They gave her a microscope, much like one of these right here. She sat down, she showed them she knew how to use it, she got that job. So you never know when something like some small thing like this might pay off. It's been good talking to you. A lot of fun teaching you how to use the microscope. See you on the flip side.
Cause my love deserves to be free. I never want to look at a house in the garden. I never want to lock you down. I know you're not mine, it's just my turn, but we can still have fun.